Hi, my name is Blake Miller, a student biology major at Augsburg College. I'm here today to convince you that three scientists, with Osamu Shimomura, Roger Tsien, and Martin Chelfie, deserve to be recognized as some of the most important scientists in cellular biology discoveries. I'm also here to inform you about what GFPs are and how they are infinitely important in uh, gene expression and uh, countless other things in the scientific um, community. What is a GFP? GFPs they're first isolated from a jellyfish, A. victoria, by Osamu Shimomura. How he did this was he would squeeze, or essentially, it would squeeze out these GFPs from the jellyfish, essentially killing them every time, which is immoral, or which may seem immoral, but as I go on, I will show you that the discoveries are way more important, or are worth this immoral sacrifice. Um, so they were first isolated from this jellyfish by Osamu Shimomura in 1962. What it is that it's a small protein that fluoresces when exposed to the light in the blue to UV range. Its major excitation peak is at about 400 nanometers, which is below that of visible spectrum. But the reason we can see it, as I will explain later, is that it can be seen under blue light, which is contained in the actual jellyfish. The GFP gene can be introduced into new organisms and have since been modified and are infinitely useful in understanding protein targeting and gene expression. So its structure, it's basically a chain of 238 amino acids, which consists of a beta barrel with alpha helical caps. It weighs approximately 27,000 grams per mole, and only about 4 of these amino acids, which is called the chromophore, directly produce any fluorescence effect. The barrel structure keeps the chromophore away from the solvents. Why it glows? The reason it glows, or the reason that it has such a green hue, is because the jellyfish have another molecule within it called the aquarin, which gives off blue light. This blue light is then absorbed by the green fluorescent protein, and at its secondary excitation peak, which lets off a green hue. The reason GFP is used in other organisms in vivo is essentially because one man, Martin Chelfie, had the idea to use GFP as a marker that could be attached to a promoter. He had hoped that the GFP, if it was produced, whenever this GFP was produced, if it was attached to the promoter, the GFP would be produced whenever the promoter was activated, and so it would be shown in genes of different organisms. He incorporated the GFP into an E. coli, and they fluoresce green when shown under blue light. This opened the doorway for viewing gene expression in a whole new way, such as mice with GFP genes introduced into their genomes, as you can see. GFP is especially useful because its folding occurs automatically without the assistance of enzymes. Um, Also, this process only requires atmospheric oxygen to fluoresce, and organisms can be genetically engineered to have genes that produce GFP. And it will still work with and it will still work without multiple genes being implanted. There are, however, many problems with this wild-type GFP found from the A Victoria. One problem is that it has broad excitation peaks, or that is, they absorb multiple colors of light, and also this makes it unsuitable for FRET, which we will discuss later in this demonstration. It also isn't slow in the formation of a chromophore. It takes about 2 hours for it to fully oxidize, and has a tendency to form dimers and trimers, increasing its molecular weight. This can inhibit the function of both the GFP and the attached protein. It inhibits the movement of TAG proteins around the cell and through the membrane. It also is non-optimal for codon usage in mammals and has poor folding efficiency at room temperature, or above room temperature, The solution was thought up by a scientist named Roger Tsien, and when he engineered colonies of bacteria to express the GFP gene and just let them grow. This, he knew, would produce many mutants naturally, and although these mutants, many of these mutants had a mutation that destroyed fluorescence, some helped improve some of the main GFP problems, such as shifted emission peaks, change in color, increased brightness, and they also oxidize much faster. These are examples of the helpful mutations he got. In this diagram, first of all, MRFPI, or MRFP1, is a previously altered protein. And so that's where these are derived from, most of these. Also, EXC right here stands for the wavelength at which it's excited at, and EM stands for the peak emission wavelength. Mutants with the letter E before the name, this says that these are enhanced brightness. And how has this helped us? Well, GFP has been infinitely helpful in many scientific fields. It is widely used in genetic studies to indicate the expression of other genes. In both pro and eukaryotes, GFP is expressed in the inhabitant, but only when the original was expressed. FRET can also be used to view real-time events within the cell by using two different GFPs that emit light at different wavelengths when the proteins interact. And so, when the proteins interact, such as an enzyme acting on its substrate. In conclusion, these scientists deserve more recognition for what they have accomplished. These accomplishments have been and still are being used to make new discoveries, expanding our knowledge of, in almost every field. The discovery and development of GFPs have been essential and still continue to be essential in the discovery and understanding of what we know about gene expression. What we know about gene expression, as well as what we know about intercellular systems, protein folding, protein transport. They have also been helpful in understanding brain circuitry, membrane potentials and have been helpful in gene therapy. Recognition could be the next step in providing more funding to continue this research and continue expanding our knowledge in every scientific field, especially biology. Any questions?